Well, good afternoon. Welcome to Room C. My name is Dar Hines. I'm a postdoc at the Center for Regenerative Medicine at Boston Medical Center. And today I want to present my work on Notch activation during mesoderm induction and how it modulates the emergence of T and NK cells from human-induced pluripotent stem cells. So here I'm showing you my full poster, but I actually broke it up so we can all see as we move through this. So by way of introduction, induced pluripotent stem cell-derived CAR T therapy has many potential advantages over conventional CAR T therapy. This would include the ability for precise germline gene editing to introduce chimeric antigen receptors, reduce immune rejection, and introduce safety features. The controlled differentiation process also allows for better quality control, potentially unlimited numbers of cells, and may even make an off-the-shelf T-cell therapeutic possible. However, it's very difficult to make a large number of T-cells from iPSCs at this point, and that will be required for iPSC-derived CAR T therapy to be possible. This is because most hematopoietic progenitors from iPSCs end up as lineage-limited primitive cells that lack T potential. Notch1 is known to be required for the production of the definitive hematopoietic stem cell, and thus we hypothesized that activation of the Notch pathway during our differentiations would drive access to the T and NK cell lineage. So to test this hypothesis, we created 4 TET-on NICD1 iPSC lines by inserting a TET-on construct driving the expression of the Notch NICD1 into the human AAVS1 locus on our iPSC lines. Here in A, I'm showing you a schematic of our targeting vector, and B, our nucleofection protocol. After pyrimycin selection and isolation of positive clones, we tested each clone by giving it doxycycline at several different concentrations, and then looking at Notch target gene expression after 72 hours. And here I'm showing you a nice dose response curve for HES5 expression in all 4 of the lines that we made. After validating these iPSC lines, we used an in-house optimized hematopoietic progenitor differentiation protocol to determine when to activate the Notch pathway to actually improve our T-cell output. This protocol produces a hematopoietic progenitor at day 12 that is then put into a co-culture system with feeder cells that are expressing the Notch ligand delta 4, and that system drives their T and NK cell commitment. So this first panel under B here is showing you early T and NK cell commitment of our hematopoietic progenitors as measured by CD7 expression at day 19, which is 1 week of co-culture, with a range of times for when we treated them with doxycycline. And interestingly enough, when we treat with doxycycline from day 0 to 2, which is during our mesoderm induction phase, you can see the most robust increase in the CD7 population at day 19. And the next 2 panels, I'm actually showing you the same conditions, but now looking at this progenitors at day 12, so 1 week earlier, and seeing if there are any markers of progenitors that would sort of read out their ability to make T cells 1 week later. Interestingly, cells negative for the erythrocyte marker CD235A, actually that population did kind of read out their T cell potential. However, more classic progenitor markers such as CD34 really did not predict their T cell potential. Then we took all 4 of our NICD1 lines and differentiated all of them using this dox treatment from day 0 to 2. And we're able to show a very robust increase in CD7 commitment by day 19 in all 4 of the cell lines that we created, showing that it's very consistent across multiple iPSC cell lines. So to approach a more mechanistic understanding of what's happening during this early Notch activation, we compared the gene expression between all 4 of our NICD1 lines that we treated with doxycycline and all 4 of their parental iPSC lines also treated with doxycycline but did not have the construct in them. We looked by qPCR at a panel of hematopoietic progenitor genes. And really the key piece of data that we collected here can be seen in this blue curve for HES5 expression between the NICD1 and parental lines. And you can see that by day 4, the expression of HES5 is equal between the two, suggesting that the parental iPSC lines turn on Notch during their differentiation. To continue on this, we sorted day 3 cells that were treated with doxycycline and not for the hemogenic endothelial progenitor population, which we identified as KDR positive, VE cadherin positive, and CD34 positive, and compared the Notch gene expression between that population and our non-mesodermal cells in the same cultures, which were KDR minus. And here we saw a very strong Notch signal in all cultures, typified by Notch 4 and Delta 4, but it was potentiated in the dox-treated cultures. So this suggests that Notch is a key pathway in mesodermal patterning toward hemogenic endothelium in all of our cultures, but the NICD1 lines allow us to turn it on earlier and potentiate this response. Our CD7 cells do differentiate out to CD8 single positive T cells by day 63. They have gene expression of a panel of T cell related genes that are very similar to primary human thymocytes, suggesting their identity is correct as T cells. And if you take our day 40 T cells and we stimulate them with either tetramers or PMA and ionomycin, we can see a nice upregulation of activation markers CD69 and 25, and also an increase in proliferation as would be expected for mature T cells. We did notice a strong phenotype in several of our cultures between day 19 and day 26. And this was the developing cells that we thought were T cells would suddenly lyse the feeder cells that they were growing on. And flow cytometry analysis would then show a very strong upregulation of the CD56, an NK cell marker, and actually loss of CD5, our T cell marker. After many culture modifications, we found that the density of our progenitors in the co-culture system had a strong impact on the T versus NK cell decision, with 50,000 cells plated leading to a strong CD5 population at day 34, whereas 250,000 progenitors would lead to almost no CD5 cells, and these same cells would stain very strongly for CD56. So at least in this co-culture system, density is very important in the T versus NK cell decision. So to further look at our differentiation process from day 12 progenitors out to day 42 T cells, so we undertook single cell sequencing at 5 different time points compared with freshly isolated postnatal human thymocytes. And here I'm just focusing on spring plots of our day 12 progenitors. And I've separated out the dox-treated and untreated cells just so you can see them more easily. And here on the left, there's a composite expression of a hematopoietic progenitor gene list, which I'm showing you over here. And you can see that the dox-treated cells have much stronger signal of this gene list. And then on the right, I'm showing you the same spring plot, but now highlighting a much more familiar marker, CD34. If we look at this bright progenitor population here, about 2,200 cells were present in that population, and about 87% of them came from the dox-treated group, and only about 13% came from the untreated cells. So almost a 6-fold increase in this population of sort of putative multipotent progenitors. And here I'm showing you our spring plots from the whole time series combined. And in panel A, I've annotated each of our clusters from progenitors differentiating out towards iPSC-derived T cells and NK cells. And thankfully, our iPSC-derived T cells did cluster close to our primary cells, suggesting a at least similar identity. In B, I've color-coded the clusters by time point, so you can also see there's a nice temporal differentiation towards the T cell fate. You can also notice that our progenitors do make erythroid, myeloid, and macrophage populations, that those just don't persist. And then in C, I'm showing you again just kind of this composite gene expression signature of a progenitor gene list, a T cell gene list, and an NK cell gene list. And you can see nicely how the progenitor sort of gene expression program comes out and then turns off right about the time the T cell program turns on. All right, there's obviously much more we could say there, but we're about out of time. So my work outlines kind of a simple 2D differentiation that efficiently produces T and NK capable progenitors across multiple iPSC lines with some clear clinical applications for possible therapy in the future. And then more specifically, notch activation during early mesodermal development yields a robust increase in T and NK capable progenitors across multiple iPSC lines. Our iPSC-derived T cells do mature into CD8 single positive cells that seem to act normally. And our iPSC-derived progenitors make this T or NK cell choice primarily dependent on density. Our single-cell RNA sequencing showed a 6-fold increase in our progenitor population at day 12, and these cells did progress towards T cells that express a gene expression program quite similar to human primary thymocytes. So with that, thank you for listening, and I'd be glad to answer any questions.
Why do you think there's a preferential differentiation into CD8 cells? Does that tell us something about the ontogeny of T cells in general?
This is actually a really good question. In all iPSC-derived differentiations that I'm aware of that are published to date, all of them see this CD8 bias to the T cells produced. I think at this point we can only conjecture that this is somehow a default process, but I will say that the selection process that's happening here, where normally a T cell will go through a positive and a negative selection process in the thymus as it matures, and this process in our culture system is probably very aberrant, might be the right word, very different than what happens naturally. And I think that something about the way they navigate the selection process must actually be potentiating the T cell or the CD8 fate rather than the CD4.
Can I follow up?
A lot more. Go ahead.
Can I follow up with David's question? So with that said, have you tried like spiking with different peptides to maybe see if you can select for or against or drive differentiation in a certain manner?
Yeah, that's a really interesting question. So the answer is no, I haven't. The experiment that I've been trying to successfully do is to take 2 different chimeric antigen receptors where we understand the intensity of the interaction between the receptor and its ligand, and see if the intensity of the signal that the cell is receiving through the antigen receptor actually influences the CD8 versus CD4 choice. So, short answer is no. I'm trying this kind of affinity-based experiment currently.
Are these functionally suppressor CD8 cells or cytotoxic CD8 cells?
Yeah, so actually I need to do more work on the sort of like characterization side of things. So right now I'm putting chimeric antigen receptors in, and I have some cell lines to target. I have a HER2 CAR, and we're going to see if it will kill some, breast cancer and ovarian cancer cells. I would say from previously published literature, most of them end up as cytotoxic and not suppressor, but I don't have hard data on mine.
One last quick question, and that is, what is your relative efficiency compared to trying to derive T cells from hematopoietic stem cells?
This is also a good question. So I haven't done an experiment going from like cord blood CD34s to T cells. What I will say is that I start with about 100,000 iPSCs at the beginning. I'll make about 1 million CD34 cells. And it seems to me that on a good differentiation, when we get out to the CD8 positive cells, I have maybe 1.5 million, something like that. So we definitely lose cells along the process.
So can I ask where the bottleneck is, or where do you think it is? Is it actually the T cell or lymphoid differentiation, or is it something upstream?
Again, a little bit hard to like nail it down perfectly with my data, but I think it's actually the selection process is where we lose most of the cells. They hit double positive cells usually quite easily, But the number of cells that seem to make it through that double positive, and when I say double positive, that's CD4 and CD8 together, the number of cells that make it through that sort of checkpoint into a CD8 single positive cell seems to drop pretty significantly. And we know from normal thymic development of T cells that that's when selection is happening. So that's what I think the bottleneck is.
Great job.
Thank you. Thank you. I think we're going to move on to poster number 6. Lillian, if you're ready. Thank you.
Great. Hopefully you guys can see my my screen there. So I am tremendously excited to share our research into how COVID infection impacts immune cells in the placenta during pregnancy. I am a second-year pediatrics resident in the Boston Combined Residency Program at BMC and Boston Children's, and this work has all been done at BMC. So I similarly am showing you my whole poster here, but I'm going to zoom in on each section so we can see the text and the figures a little bit more clearly. So this project is focused on understanding the immune response to COVID in the placenta in pregnancy. And the reason this work is important is because, as you know, COVID has really taken the world by storm over the last year. And as a result, COVID-19 is incredibly common in pregnancy, particularly at Boston Medical Center. We've seen a lot of admissions where women have tested positive for COVID, either at admission or during their pregnancy. And we now know that COVID infection during pregnancy does impact the pregnancy outcomes, both for the mother and for the baby. So on the right, I'm showing a graph that summarizes several different studies, which shows that when women had COVID-19 during pregnancy, they're more likely to have mortality than women without COVID-19 and ICU admissions. And their infants were more likely to be born preterm than pregnancies without COVID, and babies are more likely to be admitted to the NICU. So it's important during pregnancy, and it's been very common in the last year. However, transmission of the virus from mother to infant is incredibly rare, particularly vertical transmission, which is when transmission occurs during pregnancy. So this indicates that there's that the immune response during pregnancy is sufficient to protect the baby, the developing fetus. So we hypothesized that the placenta forms an effective protective barrier against vertical transmission of COVID-19, and then further hypothesized that this effective barrier likely involved the endogenous placental leukocyte populations, or the white blood cells. So as I'm showing here in this diagram, the maternal-fetal interface is pretty complex. So on the left, I'm showing the chorionic villus as it interfaces with the decidua basalis, which is on the maternal or uterine side. And there's multiple specialized placental cells that are involved in that barrier. And some of those are certainly important for the immune response to viruses like COVID. But what I'm going to focus on are the immune cells, natural killer cells, and macrophages that are maternally derived and are present on the decidua basalis at the maternal-fetal interface, and are known to be important for defense against viral infections in general, and then specifically against viral infections that occur in pregnancy and preventing transmission to the fetus. And the question we asked were whether these cells, natural killer cells and macrophages, whether they respond to SARS-CoV-2 infection in the placenta specifically. So our objective was to examine the relative abundance of maternal macrophages marked by CD14 and maternal NK cells marked by CD56 in human placental tissues in pregnancies, comparing pregnancies where COVID-19 occurred with contemporary controls without COVID-19. So now moving on into the methods. This study used human tissues that were obtained from mothers who were admitted to Boston Medical Center who were either positive for COVID by the nasopharyngeal PCR or were negative. And these were collected in the spring during the height of COVID admissions, and the women were all symptomatic. Placentas were obtained following delivery using just routine care, and they were banked and then made accessible to researchers. We obtained full thickness biopsies of the decidual tissue, formalin fixed these tissues, and then had them cryosectioned by a company here in Boston or in Salem, and then performed antigen retrieval followed by primary staining for macrophages using antibodies against CD14 or NK cells with antibodies against CD56. And then use secondary antibodies that were fluorescently labeled in order to visualize the location of these cells. We then acquired immunofluorescence images using automated microscopy, so all the images were obtained using the same settings, and then quantified the fluorescence using a corrected total cell fluorescence and creating a fluorescence ratio. And all of these analyses were performed blinded, so we did not know which groups the images corresponded to. And they were performed with 5 images per slide or placenta. And now we'll move into our results. So first, looking at macrophages, this graph, these 2 images are representative images showing CD14 staining in red and DAPI staining, which highlights nuclei in blue at 200x magnification. And on the left, I'm showing a representative image from a placenta from a COVID positive versus on the right, a control sample. And I think it's very easy to appreciate that there are a lot more macrophages present in the COVID-19 positive sample. But to do this in a more quantitative manner, we quantified the fluorescence in these images and compared the total fluorescence ratio in the COVID-19 positive samples compared to control, shown in the graph here on the left. What we found is that there was significantly greater fluorescence ratio in those COVID-19 positive samples by an unpaired t-test. And this is less than 0.01 in significance. Moving on to the NK cells. Here again, I'm showing the COVID-19 image on the left and the control on the right. And in this case, the marker is CD56, which was already introduced as an NK cell marker in the previous presentation, as well as the DAPI counterstain. And here the findings are a little bit more subtle, but I think you can appreciate that there is a little bit more of those red stained cells in the COVID-19 positive sample compared to the control. And again, when we quantified this using the fluorescence ratio, there were significantly more NK cell fluorescence in the COVID-19 compared to the control samples. And this was significant using an unpaired t-test at 0.05. Although I think you can appreciate that the quantity of the fluorescence ratio is less significantly, is more similar compared to the macrophages where there was really a pretty drastic difference. So to summarize, we found that NK cells and macrophages are both accumulate at the maternal fetal interface in pregnancies that were complicated by COVID-19 compared to contemporary controls. And so this is really preliminary and we can't really talk about causation in this study, but it suggests that NK cells and macrophages might be important in the placental response to COVID-19, potentially in preventing vertical transmission, but also potentially in driving inflammation that may have adverse outcomes like preterm birth or other complications of pregnancy. So I think that these findings are incredibly exciting and show that COVID does have an impact on the placenta, but there's really a lot more to be done. So as part of this project, we are now in the process of obtaining samples from maternal fetal dyads where mothers had documented COVID during either their first, second, or third trimester, and comparing those to control maternal fetal dyads, so we can understand what's happening both at the placental level and in the baby. And our goal is to both look at more immune cell infiltrates like T cells and neutrophils, as well as do cytokine profiling to really understand the inflammation at the placental level. This will help us understand the immune response to COVID and in the placenta. And what I'm particularly interested in is comparing how that immune response differs based off of the timing of maternal infection during pregnancy. So for instance, whether a first trimester infection leads to prolonged and lingering inflammation throughout pregnancy versus complete resolution of inflammatory findings if the infection occurs earlier in gestation. So with that, I just want to thank you for listening. Thank the BU for the CTSI grant, which was essential for this work. I'd also like to thank the Fred Lovejoy Residency Research Grant that I obtained through my residency program, all of the staff members in labor and delivery, in the NICU and in the newborn nursery for helping with sample collection, and then all of our collaborators. With that, I am happy to take any questions.
Have you had a chance to collect RNA from the placentas to see whether or not there are signals for other cells besides the NK cells and macrophages?
Yeah, that is something that we would really like to do. We have samples that we could perform RNA-seq analysis on. It's just really a matter of having the funding to be able to go forward with that. So that is something we'd be very interested in doing. I'm hopeful we are moving forward with a cytokine analysis that may give us some clue to what other cells may be present. That's kind of our next step, which we'll be doing in the next month or so. But I would love to do RNA-seq to or even just RT-PCR to get a sense of all the cells present.
So I have maybe a really general question. Are there other examples of maybe diseases where you have adverse birth outcomes and people have tried to characterize cell populations and did they see similar things, or is it very specific? So I guess I tend to think about HIV a little bit. So maybe like HIV, I know that there's a lot of preterm issues with birth. Do you see similar increases in inflammatory cells, for example?
Mm-hmm. So that's a great question that I should know the answer to, but I can't give you a great answer off the top of my head. So certainly we do see inflammatory responses to other viral infections and also bacterial infections during pregnancy in the placenta that involve macrophages and NK cells, as well as other cells like lymphocytes and neutrophils, kind of depending on the chronicity of the infection. But I'm not certain how that correlates with outcomes and with the, for instance, with vertical transmission or adverse fetal outcomes. So that's something I'll have to read into more.
Okay, sounds like we're good. Thank you. So we can go on to poster 14.
Yeah.
Thank you.
All right. Let me just get on the laser. All right, hi everyone. Yeah, so my name is Rhiannon Wetter and I'm a postdoctoral fellow at the Center for Regenerative Medicine. And today I'm going to be telling you about my project, which is looking at risk genes associated with chronic obstructive pulmonary disease and what these risk genes may be actually doing in type 2 alveolar epithelial cells. So chronic obstructive pulmonary disease, or COPD, is a chronic devastating disease of the lungs, which results in the loss and destruction of the distal alveolar units, and this is termed emphysema. And these distal alveolar units are lined by two cell types. They are lined by type 1 and type 2 alveolar epithelial cells. So we're particularly interested in type 2 alveolar epithelial cells, and these cells produce surfactant proteins which relieve pulmonary tension as we breathe. And in addition, they also act as the facultative progenitors of the distal lung in that they can self-renew and also transdifferentiate into type 1 cells, which are then the cells responsible for gas exchange. And so we think that a loss of type 2 alveolar epithelial cells over the course of disease could be really important in disease pathogenesis of COPD. So in addition to extrinsic factors such as cigarette smoking, we also know that there's a really strong genetic component to COPD inception. And one really strong key piece of evidence that we have for this is that we know that low lung function in early adulthood actually precedes COPD development, which normally happens much later in life. And then, so this suggests to us that there is at least a subset of patients with COPD who never developed fully functional lungs in early adulthood. And this is probably again, there could be extrinsic factors at play here, but there's also a really strong genetic component to that. And so this genetic susceptibility, we've seen evidence also by GWAS, and we see overlapping GWAS risk alleles between both COPD and low lung function. However, to date, we haven't really been able to understand what these COPD risk genes actually do, and especially not in type 2 alveolar epithelial cells. And so that leads me to my aim, which is to try to understand what these lung function risk genes do in type 2 alveolar epithelial cell differentiation and also function. So primary human type 2 alveolar epithelial cells are very hard to access. They sit in the very distal regions of our lungs, so we can't really gain access to them particularly easily. And even when we can, for instance, through explant lungs, the ability to maintain human type 2 alveolar epithelial cells in culture is really quite limited. And so instead, what we chose to use was human-induced pluripotent stem cells, which we can then differentiate to type 2 alveolar epithelial cells. So the iPSC lines that we are using are patient-derived, and we have targeted these lines with specific reporters that allow us to read out how our directed differentiation is going. So for instance, we have a GFP reporter that reports for NKX2-1, which is an essential transcription factor in lung progenitors. We also then have a reporter for SFTPC, which is only expressed in type 2 alveolar epithelial cells. So when we see this reporter turning on, we know that we're making type 2 alveolar epithelial cells. In order to interrogate gene function, we decided to use a CRISPR interference system. And to do this, we targeted a tetracycline-inducible DCAS9 crab to the AAVS1 locus, which is a safe harbor locus in the human genome. And then we confirmed appropriate integration of this construct there. In addition, though, to actually get gene knockdown, we need to introduce guide RNAs targeting our genes of interest. And so to do this, I either in vitro transcribed guide RNAs, or I could clone multiple guide RNAs targeting the transcriptional start site of a gene of interest. And I could then deliver this to the cells using a lentivirus. And lastly, for my methods, I just want to talk you through a little bit about directed differentiation protocol to make type 2 alveolar epithelial cells. So in this protocol, what we do is we take cues from the developing embryo and we try to mimic this in the dish. So we start with iPS cells, and we can then derive definitive endoderm, which is one of the 3 germ layers in the embryo. We can then go on to make anterior foregut endoderm, as well as then onto lung progenitors. And these lung progenitors express NKX2-1. We can then sort for NKX2-1 positive cells and replate them in media that then promotes differentiation to type 2 alveolar epithelial cells. And as I mentioned, a major caveat of primary type 2 cell culture is that the cells don't survive particularly long. However, our iPSC-derived type 2 cells, we can passage and maintain in culture, and that then allows us to actually do a lot more and interrogate the genes that I want to interrogate. All right, so moving into figure 2, and this is where I'm characterizing my CRISPR-I targeted iPSCs. So in panel A, what you can see is a schematic of how CRISPR interference actually works. So as I mentioned, I have this dCas9 crab construct, which has a tetracycline element, and this is stably transduced into the cells. So that means that when we add doxycycline to the cells, this turns on dCas9 crab expression. This dCas9 crab can then be guided to the transcriptional start site of our gene of interest, and that's through the guide RNAs that I've introduced. And once it's bound, it then blocks gene transcription. So it's a little bit unlike conventional CRISPR, where you would have cutting of the DNA and potentially indels, and instead this just blocks gene transcription. So as you can see, within 24 hours of adding doxycycline to the cells, we have dCas9 crab beginning to turn on. And then as sort of a sanity check for myself, I wanted to ensure that we could knock down genes throughout the directed differentiation process. So I started off by just knocking down a key transcription factor in iPSCs, and this gene is called POU5F1. And as you can see, introducing the guide RNAs knocks down both the mRNA of POU5F1 as well as the protein level. I also wanted to check that I could knock down genes in lung progenitors, and so I chose just to look at NKX2-1. Again, you can see we can efficiently knock down NKX2-1. And likewise, I wanted to check that I could knock down SFTPC in type 2 cells, and we can indeed achieve this. So now I felt like we had built a model system where we could now interrogate genes of interest that are associated with COPD. So what I'm going to show you today is data from 9 genes that we've looked at knocking down, and these genes are shown here. And what I want to really note to all of you is that we really don't know to date what these genes do in type 2 alveolar epithelial cells, or to be honest, for some of the genes, there's very little known about them, full stop. So it's a really exciting model that we can then try to tease apart the gene function of these particular risk genes. So what we're looking at in this panel is knocking down these genes of interest in the lung progenitors. And so these lung progenitors, as I mentioned, now go on to differentiate into type 2 alveolar epithelial cells. So we want to see if loss of these genes affects the differentiation and derivation of type 2 cells in the first place. So in panel B, we're looking at SFTPC expression, which is only expressed in type 2 alveolar epithelial cells. And you can see that knockdown of various genes has differing effects on SFTPC expression. So for instance, you can see that knockdown of FAM13A, which is shown here in purple, or knockdown of MFAP2, which is shown here in yellow, decreases expression of SFTPC. And so this could be either that we're deriving fewer type 2 alveolar epithelial cells, or the type 2 cells that we are deriving just cannot upregulate this really key protein within themselves. On the flip side, you can see that knockdown of DSP or knockdown of HIP actually increases SFTPC expression, suggesting that in normal development that these genes are actually repressing type 2 cell derivation. You'll note I also always include NKX2-1 as kind of just a sanity control to know that we're still maintaining a lung identity, and you can see that there's no significant difference in NKX2-1. Lastly, we wanted to look at what these genes were doing in already established type 2 alveolar epithelial cells. So as you can see, again, we could successfully knock down these genes of interest. And then the first readout that I looked at was proliferation of type 2 cells, because we know that especially in injury, these cells need to be able to proliferate in order to self-renew and also repair the alveolus. So I looked at proliferation by looking at EDU incorporation into the cells. And again, we saw differing patterns depending on the gene being knocked down. So for instance, FAM13A knockdown decreased proliferation of these cells, whereas knockdown of DSP, RBMS3, TGF-beta2, and a gene called ADGRG6 all increased proliferation of type 2 alveolar epithelial cells. And you can also see that knockdown of some of the genes had no effect. Likewise, we then looked at maintenance of SFTPC expression, and again, you can see some interesting patterns emerging. Again, there's some of the same genes popping up: FAM13A, DSP, RBMS3, ACHIP, ADGRG6, and SOX4, all significantly increased SFTPC expression. Again, no major changes really with NKX2-1 expression. And then the last gene we looked at was SFTPA1. And we've shown previously that this gene is associated with maturation of type 2 alveolar epithelial cells. And we actually see a really similar kinetic to what we see with SFTPC expression. So what I've shown you today is that we've been able to establish an iPSC-based CRISPR interference model in order to temporally control gene expression. And then using this model, we've been able to interrogate gene function of these genes that are associated with COPD and lung function, and understanding what these genes do in the derivation of type 2 alveolar epithelial cells and also in already established type 2 cells. So in future studies, we want to start to now use this platform to combinatorially knock down multiple genes of interest and see how that may have a synergistic effect. And lastly, we're really now delving really deeply into some of these genes to understand the molecular mechanism by which they control type 2 alveolar epithelial cell function. Oh my goodness, I'm tongue-tied. Thank you. Love to hear any questions. David?
Tour de force, a truly fabulous sequence of events. And let me ask you what readouts you're going to look for to determine whether or not there are alterations in type 2 cell function, and whether or not you have any plans for beginning to see whether or not there's reversal of dysfunction with commonly used drugs or other experimental potential treatments for COPD.
Yeah, that's a great question. So to answer the first part of your question, I think we're already starting to get at some of the type 2 cell function in terms of looking at surfactants, looking at their capacity to proliferate, but there are some key ones that I didn't mention. So for instance, their ability to make type 1 cells, which is a little bit limited in our laboratory setting at the moment, but it is something that others in the lab are working towards. So I think that will be something we'll be able to do quite soon. And then I think the other major readout would be response to injury. So we have plans to smoke these cells, you know, with and without gene knockdown, and see how they respond to that sort of thing. So I think that will give us the best overall sense of type 2 cell function. And then to answer your second question, that's not something I've really actually thought about yet, but I think again, that's definitely a really great idea. And I think especially like in that injury context, I think that would be really, really great to see whether loss of some of these genes, but not others, has effect on drug response.
So I like the idea of doing combinations, because I'm guessing that that'll tell you a little bit about complementation, or if things are in, you know, unique pathways. One sort of technical question, and I just couldn't kind of line up all the lines and squares. Was the robustness of your response anywhere related to how easy it was to knock down maybe your target genes? Yeah,
great question. And that's something I still grapple with. It's very challenging to really tease that apart, but I think the answer is yes. So I've started to do some studies, trying to kind of toggle the gene expression knockdown, because kind of, you know, initially I was just like, knock it down as much as possible to try and get at gene function. But now, I'm trying to play with like I can play with doxycycline, for instance, or I can play with how much guide RNA the cells have. And definitely it does seem like the robustness of the response is proportional to the level of knockdown.
And I know these were candidates from a GWAS screen, but are these like ones that are very common, or did you sort of randomly pick these out because you knew they were part of the differentiation pathway, so they were kind of low-hanging fruit, or just kind of that jump?
Yeah, great question, and I did not explain that well at all. Some of these genes are basically like the top hits that come up in every COPD GWAS study, but we also had to ensure that these genes are expressed in type 2 alveolar epithelial cells. So we did look and ensure that the genes that we were picking were expressed in both primary type 2 cells and in our iPS-derived type 2 cells. So there was a little bit of cherry picking, but they are, we did ensure that the genes are reproducibly shown across many GWAS to be associated. And some of the genes like HIP, for instance, and FAM13A are like the biggest players in COPD GWAS at the moment. So yeah.
How uniform is the response across donors of the iPS cells?
Yeah, pretty uniform. For me, actually, the limiting factor is how well they differentiate in the first place, which is there is line-to-line variability in terms of how well the differentiation goes. But once I can get over that hurdle, and I see very reproducible results.
Do you have plans to study people who iPS cells and TUF2 cells derived from iPS cells from individuals, some smokers who never get COPD, compare those to those who obviously have the disease?
Yeah, we actually do. That's kind of a long-term goal of the lab, and we have banked samples from the Framingham Heart Study of long-time smokers who never developed COPD, those who are sort of age matched and did develop COPD, and so that will be really interesting in the future to look at. Great, thank you, everyone.
Thank you. Okay, so we're going on to our final posters, poster 30, and Divya, if you're ready.
Yes. All right, I will just share my screen. Does everyone see my screen?
Yes.
Okay, great. So hi everyone, my name is Divya. I'm a second year medical student at Boston University. My project is on co-segmentation of glomeruli on a number of histological stains using deep learning. So just as a brief background, kidney disease is very prevalent in the United States. An estimated 15% or 1 in 7 adults in the US has chronic kidney disease. And this is according to the CDC. In order to help identify the underlying cause, renal biopsies are often obtained from kidney disease patients as part of the workup. So during the histopathologic assessment, pathologists routinely examine multiple sites of a renal biopsy to assess the grade of kidney disease and provide a diagnosis. And this histologic assessment includes glomerular counting and estimation of glomerular sclerosis. This process of counting the glomeruli and comparing them across the stains, though, can be very tedious and cumbersome for the pathologist. With advancements in machine learning, glomerular counting and estimation of glomerular sclerosis can be greatly improved in terms of accuracy as well as efficiency. So in terms of the objectives for this project, our first objective was to use the digitized renal biopsies to develop a patch-based deep learning model. I'll briefly explain what I mean by patch-based later on. But in essence, we're creating a model to perform glomerular segmentation, which is essentially outlining of the glomeruli on the biopsies, and estimate the extent of glomerular sclerosis across the multiple histologic stains. And then finally, our objective is to essentially summarize all of this data in an automated report that contains a list of the identified glomeruli across the different stains, their corresponding locations, and the percent of glomerular sclerosis. And I'll show you a report later on just so that you get a better idea of that. So just in terms of the study population, we used 60 kidney biopsies from Ohio State University Wexner Medical Center. Some of these biopsies came from the same patient. And for each of these biopsies, we had anywhere from 1 to 4 stains. And these 4 stains were hemotoxin and eosin, H&E, periodic acid chip, PAS, Jones methanamine silver, Jones, and then trichrome. So this is just showing the details for the deep learning framework we used. We used 4 different DenseNet models, one for each stain, because each stain has different color variations and textures for the glomeruli. And we used something called a multi-class classification of the biopsy patches, which essentially divides the patches into different mutually exclusive categories. So in this case, we divided the patches into 3 different categories. No glomerulus tissue, so that's tissue that doesn't have any glomeruli, NG. Normal or partially sclerosed glomerulus, so NPS. And then finally, globally sclerosed glomerulus, GS. So just as a bit of a background, each of the renal biopsies, in terms of the digitized version, they're very big images. They are roughly 20,000 to 10,000 pixels. So 20,000 by 10,000 pixels wide. So these are very, very big images. And because of that, in order to give them as input into the models, we essentially cut them up into small pieces. And those pieces we called patches. So these patches are defined as of size 224 by 224 by 3 pixels. And finally, to train the model, we made sure that we gave a balanced data set that had a ratio of 1 to 1 to 1 for each of these categories. So just this is the schematic for the computational pipeline. What you see are the 4 different stains on the left. And the WSI stands for whole slide image. So these are the digitized renal biopsies. And from these huge renal biopsies, as I mentioned, you divide them into smaller patches. We had anywhere from 1,000 to 5,000 patches per each renal biopsy. And each of these patches are then fed into the model. So it's the DenseNet model in this case. And from that DenseNet model, you then get an annotation. So it shows you the outline of what the glomeruli are based on that model. And as you can see in the annotated whole slide image section, you can see that the green marks where the glomeruli are. And then finally, from the different annotated whole slide images across the different stains, you get a glomerular report, which shows you the number of glomeruli, the locations across the different stains, and what type they are, whether they're normal or partially sclerosed versus whether they're globally sclerosed. So here are the results for the PAS stain. Just for time purposes, I didn't show the other stains, but in essence, the values are very similar for the other stains. So what you see is on the left are the unannotated images. So these are the whole slide images for the renal biopsies. On the rightmost column, you see the pathologist annotations. So this is where the pathologist went and marked the glomeruli for us. And then in the middle are the predicted annotations made by the model. The green indicates the normal or partially sclerosed glomeruli, while the purple indicates the globally sclerosed glomeruli. And what you can see is that most, if not all, of the glomeruli annotated by the pathologist are predicted by the model. But there are some cases where it overestimates. For example, on the top region, you see that there are some more purple globally sclerosed glomeruli predicted compared to the annotation made by the pathologist. Ultimately, however, you can see the accuracy is quite high, with the accuracy of 0.977 and the kappa score of 0.667. So just for reference, the kappa score is a statistical metric used to identify the inter-rater agreement. So it's basically looking at how aligned the model's predictions are compared to the ground truth or the pathologist's predictions, annotations are. And for the kappa score, 0 is defined as no correlation between the two, and 1 would be perfect agreement between the model and the pathologist. So here are the results. It's the same thing, just in a different format, and this is for only the first case from the last slide. What you see are basically the statistics involved. So in the first biopsy showed in the last slide, there were roughly 3,000 patches, out of which almost 99% were all non-glomeruli tissue. And this makes sense, right? Because the glomeruli are only a very small portion of the renal biopsies. And what you see though is that there's only 15 actual globally sclerosed patches, and that's very, very small compared to, you know, the 3,000 in total non-glomeruli tissue. And this is one of the tricky things for the model to kind of learn is how do you deal with a case where there's 99% of one category and less than 1% of the other. And this was something that we took a lot of time into looking at. But even so, you can see the accuracy is quite high with 0.983, and the kappa score is also quite good with 0.755. So just in terms of conclusion, a patch-based deep learning framework can accurately perform glomerular segmentation in the renal biopsies. However, there's still a lot of work that needs to be done. As I said, I showed you the results for the past stain, but some of the other stains we still are working on. And then once we have the stains, the models for all the other stains fine-tuned and ready to go, we can then figure out how to generate the automated report. And then finally, there's always more fine-tuning of the deep learning model that can be worked on. So through this project, we see that machine learning can be used to minimize the time and effort needed for pathologists to evaluate the digitized renal biopsies. And, you know, hopefully in the future, this could actually be utilized by the pathologist. Right now, we're still in the research, you know, aspect of it, but eventually we want to make sure that we're able to get this technology out there. And that is it. Thank you for listening. If you guys have any questions, I'd be happy to answer it now.
Maybe I have a practical question. It's maybe a naive question. How important is the quality of the biopsy and then the staining procedure? Is there a way that you train the machine learning to compensate for that or correct for that?
So the tricky thing with machine learning is, you know, when you get the data, that's your ground truth. You can't change the quality of it. So with the renal biopsies we got, we couldn't really look at what's better and what's not. We just took all of them at the face value of they're all equally good. And I guess that's one of the downfalls of machine learning is that it's really only as good as the data it's given. Without good input, there's really no way it can do well.
Have you had a chance to take any of your AI generated data and then go back to the original pathologist who read the slides to see whether or not they agree or they disagree or they think it's valuable?
Yes, we did that. There were some instances where it would catch other blood vessels, right? Because glomeruli are capillaries. So it's a bit of an issue because, as I said, we're cutting the image into these small patches. And if you cut them into small patches, you kind of lose the context of how big the vessels are. So that was one issue that we are still looking at. But yeah, she said that it was doing well. So I'm happy.
How much more efficiently can the AI-generated program interpret the biopsies than a traditional pathologist?
So I think in terms of the model, it takes roughly 3 minutes to outline it. And as you can imagine, for the pathologist, going between 4 different slides, you know, focusing the microscope and then trying to count the number of glomeruli while going across it can take I think it's like roughly 10 minutes. It's not too much longer, but I think just like in terms of efficacy and just convenience, it's a lot better.